Welcome to the MEF2C cast, the podcast advocating for the MEF2C haploinsufficiency community by broadcasting the voices within. I'm Eli.
And I'm Haley. We are parents to a boy with MCHS and autism, sharing our journey with you throughout conversations grounded in learning, support, and community.
This is Advocacy Amplified. Do you have questions, comments, or a story to share? Want to be a guest on the show? Connect with the MEF2C cast on Facebook, mef2ccast.com, or email us at mef2cast@gmail.com.
If you enjoy the show, please support us by subscribing to the show on Apple and Spotify, leaving us a review, and following us on social media. You can also buy MEF2C cast merch at our Shopify store, linked in the podcast description. The MEF2C cast is for informational and entertainment purposes only. Views expressed are those of the hosts and guests and not necessarily of the podcast or its sponsors. This is not medical advice. Consult a healthcare professional for medical concerns.
Alrighty, hi everybody, and welcome to the MEF2C cast. We are here with Dr. Tim O'Sullivan, an immunologist at UCLA, and one of his graduate students, Cassidy Lee, who does a lot of work relating to the MEF2C gene and the immune system. How are you guys today?
Great, Eli.
Good. So can both of you talk a little bit kind of how you got involved in this specific field of like MEF2C related research to start?
Yeah, sure. So it was actually an accident. So it's like one of life's serendipitous discoveries. We're actually coming at this from a different angle. We're very interested in how different genes control the function of white blood cells of your immune system called natural killer cells. These were cells that were discovered back in 1975 that when you put them in a dish with tumor cells, they naturally kill them, thus their name. And so since then, you might have heard about T cells, right? These sort of like important immune cells that are important in response to vaccination and formation of an immunological memory. But not a lot of work has been done on human natural killer cells. And so we're really interested in the genes that regulate their function because it turns out they're very important for antiviral responses. Joey Lee, a previous MD PhD student in the lab, he did a screen of genes called transcription factors, which control the expression of genes. And he identified MEF2C as the top hit in this screen as something that was really important for human NK cell function. So again, we weren't looking for it, but we found that MEF2C was really important. We were interested in extending these findings, and it was really interesting to learn that MEF2C haploinsufficiency syndrome is actually a rare form of neurodevelopmental disorder, and this exists out in the patient cohort. But what was not known is whether these patients actually have immunological defects. And so Joey started digging into some of the clinical data and noticed that some of these kids actually have recurrent viral infections. And so that was really interesting to us because we saw in the lab, right, that these MEF2C deficient NK cells that we're knocking out with CRISPR, they have decreased functions, right? So we were actually able to collaborate with clinicians in South Carolina, and we actually were able to get samples of 2 patients with MCHS, and we were able to take the blood and then profile their NK cells, and it turns out they were also defective. So this was the first indication that a rare neurodevelopmental disorder was also an immune deficiency, which was pretty cool that we could actually predict in the lab using CRISPR gene editing that we could actually identify patients that also had similar phenotypes. So that's how we got into it.
Yeah, that's incredible. Can you, whoever wants to take this, give us just a brief overview of sort of the MEF2C, both the gene and the protein in the body, sort of from your perspective?
Yeah, you want to take this, Cassidy?
Yeah, so MEF2C was a gene that was originally found, I believe, more in muscle tissue or specifically skeletal muscle tissue as well as cardiac tissue. And then, as I'm sure you know more than any of us, MEF2C haploinsufficiency syndrome causes these really bad neurodevelopmental disorder. And so that led to, research with the gene and the protein in brain. And then there were a few case studies talking about how some individuals with MCHS have viral susceptibility or increased risk of catching viruses or having bad side effects of having viruses. And so then that's kind of what led us to have it in this screen of transcription factors, um, to see what may cause deficiencies in these natural killer cells. So, while most of the research in the MEF2 family of genes has been mostly in muscle tissue, brain tissue, we're finding that it has a really important role in the immune system, and we hope to see if not only natural killer cells, but in other immune cells such as macrophages, there are similar defects.
So when you say increased susceptibility to viral infections and you're talking about like in the literature, are you just saying like kids get sick a lot or kids tend to experience like more systemic infection, more complicated than cuz just thinking as a parent, kids just get sick a lot in general and I'm just curious as to how you tease that out from being an effect of MEF2C as opposed to just you know what I'm saying?
Sure.
Yeah.
Yeah. I think that a lot of these case studies, especially because the clinical cohort is it's pretty rare disease, right? So getting like a full statistical analysis to actually tease this apart would be difficult. But what I can tell you is that if we look at the ability of natural killer cells, for instance, to perform their effector functions, they almost look like they don't work, according to our recent publication. And so that for us is consistent with phenotypes that you would see with people that don't have NK cells, and the phenotypes that they present with are enhanced susceptibility to viral infections, specifically herpes virus infections like human cytomegalovirus
Uh-huh.
or Epstein-Barr virus. So, again, MCHS as like a neurological disorder is also relatively new as well, right? So, not a lot really is known about what happens to these kids as they grow up as well. And I
Mhm,
think obviously the neurodevelopmental disorder is sort of the most prevalent phenotype, right?
mhm,
And I think this speaks to MEF2C's role as a transcription factor, and it works not only in neurons, right, in the brain, but also in muscle tissues, but also in the immune system, right? So this is a highly complex disorder where MEF2C's role in all of these systems is highly complex. And then you get a phenotype that can also be heterogeneous in different patients depending on the type of mutation and different functional loss of MEF2C depending on where that mutation is.
right.
So it's not always gonna present the same way, right? But,
Mhm,
um, I'm part of, a foundation, or at least on the advisory board, it's called, the Rare Bird Foundation,
mhm,
and this is a collection of, um, patient advocates for kids with MEF2C haploinsufficiency syndrome. And I've actually gotten a lot of emails from parents that have told me this makes a lot of sense because their kid, when they do get sick, they remain sick longer and it's more
mhm.
severe. And a lot of children end up having with the syndrome actually have to end up being homeschooled because of the recurrent viral infections.
Interesting. I know that there are several kids with MCHS that are homeschooled. I didn't realize that that may be the reason.
Not always,
Right,
but
right, sure, of course, not necessarily, but I hadn't considered that as a possible reason. That's really interesting. So, some of your work also well, no, let's start here. So, the NK cells. So, do people with MCHS not produce as many, or do the ones they do produce not function properly, or how exactly does that work?
Cassidy?
Yeah, so patients with MCHS, I believe, produce similar amounts, but there are certain developmental markers within an NK cell. And so transcriptionally, then MEF2C, having this mutation in MEF2C alters the developmental states that this NK cell exists in. And so because of this, then the NK cells are a bit more defective or are a bit worse at killing cells as they are naturally inclined to do, or producing anti-inflammatory cytokines like interferon gamma.
So Eli, in the life cycle of an NK cell
Mhm.
and a lot of white blood cells, they'll start off in an immature state and then they'll differentiate by expressing a lot of gene programs that allow them to exert killing functions. And so we call this cytotoxicity when another cell kills another cell. And so there are genes that are associated with cytotoxic programs that have to be upregulated during maturation in order to achieve this state. And it looks like in the kids that we profiled with MCHS, they have a higher amount of these more immature NK cells, and they have a lower amount of these more mature NK cells. So they're all like altered subset differences, But then if you look at the function of the cells as well, they have really severely dysregulated lipid metabolism. So normally when you activate an NK cell with its preferred growth factors, these proteins called cytokines, they want to grow really fast. They want to proliferate, right? And in order to do that, as cells proliferate, they have to divide and they have to create from a parent cell a daughter cell, right? So one cell budding off the other. And in order to do that, you have to create an entirely new cell's worth of lipid membrane, right? Or else you can't make a cell. And in
Right.
order to do that, you not only have to synthesize lipids inside of that parent cell, but you also have to take up lipids from the environment. And it turns out that the MCHS patient NK cells, after you stimulate them with cytokines that make them grow, they don't actually increase their lipid content. So they have defective proliferation, and lipid metabolism is also very important for fueling these what we call effector functions, or their ability to kill and produce cytokines that are broadly antiviral.
Gotcha. So they're not producing the lipids they need and or processing the lipids that they're eating in order to make new cell brain membranes to produce those new daughter cells. Is that correct?
Yeah.
Okay. So maybe you guys don't does MCHS seem to affect lipid metabolism in other kinds of cells, or have you not looked into that at all? Are you only looking into the natural killer cells?
That's a great question. So we actually don't know. We've only looked
Mhm,
at natural killer cells at this point.
mhm,
I think one thing that Cassie is really excited to do is extend these observation to other immune cells like macrophages.
mhm.
But it could be that because of the central role of MEF2C as a transcription factor, it could broadly regulate lipid metabolism in other cells, like in the muscle and also in neurons.
Right.
So I think Cassie can kind of speak to what her current evidence is pointing to.
Yeah, I don't think there's much like evidence on the connecting MEF2C to lipid metabolism in other cells so far, but I do think that it would be interesting for us to take muscle cells or heart cells and to just see if lipid metabolism is affected by F2C.
Yeah, that would be because I know actually James Kelly, who I imagine you know Tim through Rare Bird. His son is on a ketogenic diet to control his
Uh,
seizures, and I know of some other kids who are on that protocol as well, and it does a good job of controlling seizures. But it would be interesting to see, what interactions that has with this lipid metabolism, because if they're not metabolizing lipids well enough, or maybe that is giving them more lipids that may help aid kind of mitigate this stuff, that might be something to look interesting to look into.
yeah, that's a really interesting suggestion. The ketogenic diet, like outside of MCHS, is also known to help with seizures
Uh-huh.
and other disorders as well. But I think that could be really interesting synergy. For instance, we have some mouse models where we have mice that are haploinsufficient for MEF2C in collaboration with Dr. Steve Skinner. It'd be really interesting to put them on a ketogenic diet and then see what happens to immune function.
Yeah. Kind of related to that, in your paper, I believe you had one model where you supplemented with oleic acid. Can you kind of talk about that a little bit and what that, what the outcomes were there?
Sure. Yeah. So one of the things that we profiled when we knocked out MEF2C in mouse and also in human NK cells is we wanted to understand what genes that MEF2C was actually controlling to express. And when you knock out MEF2C, it turns out that these transcription factors and their downstream target genes, these called SREBP family genes, 1 and 2, they were heavily downregulated, which suggests that MEF2C is driving the expression of these canonical transcription factors in lipid metabolism pathways. And so we thought, okay, if we just add back the lipids that these pathways are trying to generate, maybe we can restore the function of MEF2C deficient NK cells. And so we were sort of metabolism naive at the time. So we said, oh, look, SREBP1 pathway is down, and we can put back in maybe some of the upstream lipids that this pathway would need. Things like cholesterol. And it turns out if you put in more cholesterol, it actually turns out to be inhibitory because the cell can sense that there's too much extracellular cholesterol, and then it actually turns these pathways off. So if we take oleate, which is a downstream process, so it's converting upstream lipids into oleate, we can actually restore the function because it's missing some of the enzymes that are converting some of these precursor lipids into oleate. And so when we supplement the cells with oleic acid, now the MEF2C knockout NK cells, they can restore some of their function, and so can some of the MCHS patient NK cells. They can actually have enhanced killing function, but not increased production of inflammatory cytokines. So there, we think that the ability of NK cells to kill a virally infected cell is really highly dependent on their lipid membrane structure. They like to create these cool little projections that come out of the cell that form a cytotoxic synapse with another cell. And to do that, you need to create these lipids that are called sphingomyelins in order to make that cytotoxic synapse projection work. And so we think oleic acid is actually upstream of sphingomyelin synthesis, which is also something that Cassidy studies as well.
So what is that cytotoxic synapse? You lost me a little bit there. What is the actual function of that?
Yeah, so basically the way that cytotoxicity works is that you have to have two cells coming together. You have to actually form a synapse. And what that does is it prevents these what are called cytotoxic granules from being released into the interstitial space of a tissue and just causing nonspecific cell death. So what you want is you want your NK cell to form a close contact with its target cell and then create sort of a specific region for which these cytotoxic granules that contain proteins called granzyme B and perforin. Perforin pokes holes in the target cell, and then granzyme B goes in the cell and it starts degrading proteins, causing the cell to die. So you don't want those cytotoxic granules to just be released into the interstitial space because it'll start killing a bunch of other cells that you don't want. You only
Right.
want specific killing of that virally infected cell or that cancer cell. So in order to do that, it seems that human NK cells like to make these projections that are sphingomyelin rich in order to make those cytotoxic synapse formation with its target cell.
Gotcha. So that synapse is th- the avenue through which those chemicals that killed the cell that the viral cell or the cancer cell or whatever
Yeah,
travel.
proteins, yeah.
Okay,
Yeah,
gotcha.
and so when we're talking about synapses,
Mhm.
right? You're also thinking about neurons and neural projections, right? And
Right,
so
that's where my mind went. That's why I was confused.
yeah, but it turns out that there's probably shared metabolic pathways that are shared between neurons and also natural killer cells. So there are a lot of genes that are implied in metabolic functions that these so-called mTORopathies, or this protein that's really important in metabolism. And a lot of these patients that have mutations in those genes also present with neurodevelopmental disorders. So the process that actually takes cytotoxic granules and then gets them to that immunologic synapse is called exocytosis. And this is also something that exists inside of neuron. So we think that there also could be shared explanations for these pathways and why they present with an immune deficiency and also with neurodevelopmental disorders because they could not have the same function in those cells, but they use a lot of the same machinery and metabolic processes to have different functions in different cells.
Right. Correct me if I'm wrong, but microglia are specialized immune cells in the brain, right? And those, based on discussions we've had with Dr. Cowan, who I'm assuming are was the people you were collaborating with in South Carolina, his lab, those seem to have a role in the neuronal pruning aspect of MCHS, which plays into the neurological aspect of MCHS. So some of those, certainly not all, but an aspect of those neurological symptoms could actually be sort of immunological at their root. Is that fair to say?
Sure. Yeah. I
Mm,
mean, I think that there could be a portion of the phenotype that is controlled by microglia. The
mhm.
macrophages that you look at in haploinsufficient MEF2C mice, it actually seems to be the opposite in K cells. They seem to be a little bit more inflammatory. So I think this also speaks to kind of the complex nature of the role of a transcription factor in one cell type versus the other. So dissecting that out, I think with mouse models, I think that's you can do that. You can delete MEF2C in specific cell types and see how that affects development.
Mhm.
Um, and you can do some, I mean, with microglia, it's a little bit harder because you can't transfer those cells. They're there since the birth of the animal, and then they don't really like to get outside the brain. So they're a little bit harder to study.
Yeah. Yeah, and just sort of in general for I think the takeaway is this is all insanely complex, and I think that maybe the general public has this idea of sort of genetics that because it's like this incredibly complex natural process and it's sort of this miracle that that means that it's elegant, and that's not necessarily the case. Like, I compare things like this sometimes to, like, when I had biostats class and I would submit an assignment, and my professor would literally be like, "I don't know how the program you gave me got what you needed. It worked, but, like, you just slap a bunch of stuff together and it ends up working, but that doesn't necessarily mean that it's a super elegant or sort of streamlined, sort of intuitive process to understand." So I guess my point is that it's really complicated to sort of unravel all the strings on this.
Well,
Um, Go
if we
ahead,
think
sorry.
about it, these are complex molecular machines that have evolved over hundreds of millions of years
Mhm.
to be able to create these complex interactions and have really fine-tuned regulatory information. So it's almost not surprising to me that like a lot of these mechanisms would be very complex and
Mhm.
why we've had to, uncover these things over the last 100 years. Our knowledge increases almost every day with the pace of science. And the tools that we have now, I think, are really good to be able to interrogate how transcription factors regulate their genes and then also how to manipulate gene expression in human cells, but also in mouse cells. So, yeah, I would say that like it requires a lot of intensive study. But I think luckily for us, like these are things that we love to do in the lab.
Yeah. So it seems like this sort of field is, well, I guess not this field, but sort of this inquiry into immunology related to MEF2C is really, really new. But what, if any, sort of practical takeaways can our audience make from all the complicated jargon that we've been throwing at them for the last 20 minutes?
Yeah, so I'd actually say that, to understand the immune system, right, there's a lot of jargon. It's almost like we're speaking a foreign language. And I think that that's on us as immunologists to try to develop a sort of framework that is a little bit more accessible to the general public, right? But I do think that there have been a wonderful series of videos. I'm probably like getting this wrong, but it's like Kierstat or something like that. I don't know if you're familiar with those line of videos.
Hmm.
It's called In a Nutshell, and what they do is they have 3D rendering of systems and how they work, and they got this wonderful British guy to voice it in a very accessible way. And they explain complex scientific phenomena in a very accessible way. And so I'm happy to share that link as well with the audience. But there's a series of these videos that can show people how the innate immune system responds to bacteria, for instance, and tells you about these complex systems, but I think in a very accessible way. So some people are more visual learners. And so I think it's also different. Like if you're just sitting with a bunch of text and you're reading about a bunch of different cells and proteins and cytokines, and it can get a little overwhelming, right? But I think if you watch some of these videos, you and visualize these processes, once you have a big picture of how the system works, then you can kind of see how all these components come in. So I think having a big picture overview of the immune system is so important because then you can kind of fine tune your understanding later on of how this works.
Cool. If you could send me that link, I'll link to it in the description for this
Yeah, happy
episode
to.
so that our audience can see it. What do you think the future of treatment looks like relative to MEF2C and immune function?
What do you think, Cassie? You're the future MD.
Well, I think with the oleic acid rescue that you mentioned earlier, then it would be really interesting to see if that would be able to help with treating some of these functions. But as you were saying, it's a very complex system. And so I would think that we would want to test these in maybe some of the MEF2C haploinsufficient mice first to see if there are any side effects that injecting a lipid into you could result in. But I think that could be a really interesting avenue. There are also a lot of advancements being made in being able to do gene editing in people. And so I think that if that ever becomes a thing that is a bit more accessible and able to do, I think that could be also another interesting avenue for treatment. Those are
Well,
the
it's already a
main
thing,
two.
isn't it? Didn't they? There was someone with some sort of liver condition or something a few weeks ago, a child that they edited their genes.
Yeah, and
So, I
that's
mean,
It's
really
it is.
miraculous.
Yeah, I mean, it's still probably decades away from being,
yeah,
something that most people can
exactly.
access, but it is something that is starting to,
Yeah, the advancements
they've
they're
done it in
making
people
is
before,
crazy.
so yeah.
Exactly.
So, on the oleic acid, and if this is too close to medical advice, please just say so, but, like, what did that dose look like? And, like, oleic acid can be found in foods like olive oil, right? If I'm correct. Do you think
Yes.
that maybe integrating foods higher in oleic acid into diets of people with MCHS might have some rescue effect, or is the dosage that you've been giving like
You know, our experiments were done in a dish, at least
okay,
in that
gotcha.
setting, we can see functional rescue.
Yeah.
We still have to do some of the experiments in animal models to kinda understand, like, what the plasma concentration of lipid would have to be, and then
Mhm.
whether that would actually be, like, a safe level to actually, like, figure out in preclinical studies. So, I don't have a good answer for you
Mhm.
on whether that would actually work in a patient. It's also complicated too because, like, dietary lipids can be utilized for so many different things, and so then how would you get that level high enough to then have a therapeutic benefit? I think what Cassie was talking about in terms of like, if you wanted like a long-term fix, there are advances with CRISPR gene editing that can correct mutations.
Mhm.
A patient had severe susceptibility to viral infections, the idea could be that perhaps that mutation could be fixed inside of a precursor cell, a stem
Mhm.
cell that makes all blood cells with a bone marrow transplant. And then you could actually now functionally have a corrected immune system in that patient. Obviously, these individuals would still have muscle disorders, they would still have neurodevelopmental disorders, but their immune system could functionally be cured if their mutation could be rescued.
Gotcha.
And not all
Yeah, well,
mutations.
and ostensibly too, some of the other treatments that are being worked on, there's a team in Boston working on treatment for MCHS. There's obviously Dr. Cowan's lab in South Carolina. So ostensibly, if they were able to treat it at the gene, it would also rescue that immune function, right? Because the gene is what's causing the
Yeah.
suppress immune function. So if they could increase the production of the protein, you would, based on what we know, that would also upregulate everything downstream.
Yeah, I mean, if you correct the mutation, right, then you're not gonna have
Mhm.
loss of MEF2C protein, you're gonna have the normal expression of MEF2C inside of the immune system, and then that could functionally restore, the immune deficiency. Of course, in order to do that, you still have to have a bone marrow transplant, which, is still carries its own form of risk because recovering from a bone marrow transplant, you're now effectively kind of depleting the peripheral immune system. So then you actually enter in a period of immune deficiency, which some people can, succumb to viral infections, for instance. So it's not always the best course of action. And I think, is only usually reserved for rare cases for kids that have pediatric cancer,
Mhm.
blood cancers, where that would actually help in the treatment and sometimes be curative. So if it's life threatening, that's usually where bone marrow transplants are involved. So it's hard to say, right? Like whether an increase in viral susceptibility, more frequent infections is considered life threatening, right? It just depends on each individual and the nature of that mutation in that patient.
Gotcha. So, what are your sort of, I guess, Cassidy, you'd be the person to answer this. What are sort of your immediate next steps relative to this research with MEF2C?
Yeah, so in Joey's paper, then he kind of showed this nice hypothesis where we're thinking that MEF2C is being regulated by a larger complex called mTOR, which is a complex that's implicated in other um, aspects such as protein synthesis and cell growth and proliferation. And so we're thinking that mTOR is what's regulating MEF2C, but we're not sure exactly how it's regulating it. And so our thinking is possibly that mTOR might be directly phosphorylating or changing the protein in a way that causes it to be more active. And so I want to see how this regulation occurs. And then downstream of it, then I'm also tackling how lipid metabolism itself through the SREBP signaling that Tim mentioned earlier directly affects NK cell function.
So there are also a lot of downstream metabolites that are controlled by the SREBPs and different pathways that we can interrogate to see not only from a basic immunology standpoint, like how are these enzymes important for NK cell function, but then also to update our knowledge of are there different metabolites that we could be supplementing that actually better restore natural killer cell function, right? We have oleic acid is sort of something we did before, but what if we understand the pathway in greater detail and there's a combination of metabolites that we can put in that now fully rescues their function, right? Kind of getting to what you're talking about. Like, is there a way of creating like a synthetic custom diet that actually could raise metabolites to a certain level that now sort of restore this really important lipid metabolism pathway function, not only in NK cells, but in muscle, right? In neurons. Could that be possible? That's something that we're really interested in. But I think using the natural killer cell is sort of like a template cell to study these because it's very easy to get NK cells from the blood of donors, right? So we can actually interrogate all these things in a very precise way with CRISPR and actually get at how these pathways are regulated in human cells without having to take muscle biopsies, right? You can't take heart biopsies and stuff like that. So we love this idea of the immune cell as sort of an ideal vessel for exploration of gene regulation that might be applicable to other cell types.
Yeah, that makes a lot of sense. Can you explain this relationship with mTOR a little more? Because like, you know, I I'm a gym rat, so like everything I hear about mTOR is related to like muscle gain. But so you're saying that it's regulating upstream of MEF2C and that it may be affect sorry, just explain that.
Yeah, no worries. Yeah, so there's a inhibitor of mTOR called rapamycin.
Mhm.
Actually, mTOR stands for mammalian target of rapamycin, and so rapamycin inhibits mTOR. And when we inhibit mTOR using this drug in human NK cells in a dish, then we see that MEF2C protein goes down. And so, because of that, then we're thinking that mTOR somehow regulates MEF2C protein, and also, yeah, mTOR does a lot of stuff. If you
Mhm.
ever look it up, it does protein synthesis, cell growth and proliferation. It's been implicated in SREBP activation before, never with the MEF2C tie, but previously it had been implicated in that activation. And so it does a whole laundry list of things.
Yeah, it
Gotcha.
turns out that the action is kind of like a black box, right? Like it's been known for a really long time in the metabolism field that mTOR is activating lipid synthesis, lipid import through activation of SREBPs. And I think there's one study that seemed to implicate how that happens, but we think that MEF2C might be the missing link, at least in immune cells, for how mTOR can actually transcriptionally activate SREBP signaling. Now, of course, we don't understand how that works, just like you. We're throwing up, question
Mm,
marks as well. But I think that's the beautiful part of a PhD. Cassidy gets
yeah,
to answer that question.
right. So, what can families and the MCHS community do to help?
Yeah. So I think that right now we're at sort of a pivotal time in science, especially in the United States. Right now, our funding climate is very restrictive and actually counterintuitive to the types of things that we should be doing. So you might be aware that the current political administration is canceling NIH grants under the guise of, DEI policies. But what it ends up turning out to be, I think, is a concerted attack on university establishment through targeting a sort of scientific studies that aren't in line with politics, right? So anything that covers vaccine development and design is basically not being funded by the NIH. There are clinical trials for people that might get life-saving therapy for cancer that are being canceled. There are people that were receiving treatments for maintaining HIV or coming up with new functional HIV cures. Those clinical trials have been canceled. And so I think that if we think about a patient cohort and a rare disease that may not be in line with drug development from big pharmaceutical companies, we have to realize that the way that we're going to get to a functional cure for a lot of these kids is that we have to invest in fundamental research because, as you know, you've asked a lot of great questions today, and we still don't know a lot of it, right? But if we don't know about it, then we're only guessing about how to get there with current treatments, right? But if we actually understand how the system works, then we can actually come up with better, more targeted therapies that probably will work better and have a higher chance of success when it gets to that clinical trial. So I think that for parents, if you want to help, definitely support these foundations like Rarebird, donate time, donate advocacy, talk to your representative in government about how important research is to you and also to your kid, and realize that, we go to work every day thinking about these things, and we don't have a political agenda. At the end of the day, we just want to understand how biology works and how can we use that to actually improve patients' lives. So I feel like we're the exact opposite group that needs to be oppressed in this moment because we literally just sort of selflessly toil in the lab to be able to get at these things.
Yeah, I agree, and I would add to that, my background is in wildlife biology, and so, like, science for the sake of science is really important because you might look at things on paper that like, studies that are being done and think, like, what good could come from that? Well, for example, there's a really popular drug used for breast cancer treatment that came from studies done with the venom from copperhead snakes, and if you just looked at, like, why are they studying copperhead venom? What good will that do? Well, eventually they isolate this chemical and it has pharmaceutical application. So you never know what kind of crossover things like that might have. So just because something doesn't have inherent value on paper doesn't mean it's not worth funding. And that's just one example. I mean, there's all kinds of examples of, things throughout the natural world where there's applications that cross fields that you would have never imagined. So,
Yeah, a great example of that, especially from Jennifer Doudna's lab, who won the Nobel Prize for her discovery of CRISPR. She was studying bacteria and how bacteria actually protect themselves against bacteriophages. And CRISPR, was this protein that was discovered that actually targeted and degraded sequences that are injected by the bacteriophage in order to help their replication. So that now, I think we even talked about it, right? Already
mhm,
has applications in people.
mhm,
And I think that we would have never found that if we hadn't actually just engaged in some form of intellectual curiosity to try to understand the natural world and then apply those, uh, learnings into sort of applied technologies. So yeah, we're very concerned that there are going to be a lot of lab closures due to cancellations in NIH funding, and we might lose really vital research. So I think that for everyone listening at home, it's really a time, I think, to be an advocate for science and realize that we're never going to get there with medicine unless we actually understand how these things work better.
yeah. Yeah, and just one more thing I would add, and we're getting dangerously close to politics here, but, like, regardless of what your political opinions are on any other given issue, you can still call your congressperson and say, "Hey, I agree with this and this and this and this, but I don't agree with what you're doing with funding whatever, funding the NIH. You're doing good on these issues, but I really think you should change your mind about this one given thing." So just
100%.
because you agree with the majority of things that a given political ideology has doesn't mean you have to agree with everything. And you can
Totally.
still advocate on this issue.
Eli, cancer doesn't care if you vote Democratic or Republican.
Right. Yes.
It affects us all equally. It does not discriminate. It couldn't care less what your political views are. And if you have a child and they end up having a neurodevelopmental disorder, it can happen to anyone.
Mm,
And again, I think us in on the science side of academics, we are not injecting politics into what we're doing. We are in the pursuit of what is true based on fact-based evidence, right?
mm,
And what we can actually hypothesize in the lab. So it's not based on preferences or agendas. It's based on the experiments that we do in the lab and using the tools to the best of our knowledge to be able to support those hypotheses. By nature, it's apolitical, right? And I think that we already know from the past the developments that have been made in medicine have already been broadly helpful, right? I mean,
mm,
you probably know someone that is taking Wegovy or Ozempic, right?
mm, mm.
Breakthrough drug that is helping a lot of people get a lot healthier in
From
response.
Gila monster venom, also another example of crossovers from unrelated fields.
Gila monster venom.
Yep.
Huh, interesting.
Yep. Well, on the note of Gila monster venom, we're going to close this down. So thank you guys so much for your time. We're really excited to hear what else comes out of your lab in the coming years. And if anybody wants to contact us, they can find the MEF2C cast on Facebook or email us at mef2cast@gmail.com. Your website is o'sullivanlab.com, correct?
That's right.
Is there anything else you want to plug before we sign off?
If you want to send Cassidy words of encouragement, go ahead and do that. A PhD is long, arduous, and I'm sure that she
It
would
would be much
appreciate.
appreciated.
Yeah. Well, the best of luck, Cassidy.
Thank you.
Alrighty,
And
thanks
thank you for
everybody.
having us.
Yeah,
Yeah,
no
thank you
problem.
for having us.